Okay, so for today, we're going to look at something, modifications, okay, to Mendelian's inheritance. So what it means by the Mendelian's inheritance, okay? So what we have learned previously, all of them, they are Mendelian's inheritance. Let's say, for example, the monohybrid cross and dihybrid cross, okay? So a quick ones to go through what we have learned previously, okay? So in monohybrid cross, okay, genetic cross. In the monohybrid cross, what we're going to have here is we only look at one gene. Okay, in this process, we'll only look at one gene. Okay, for example, in this case, we're going to look at gene that code for the seed color. Okay, so dominant trait in this case is yellow color. Recessive trait, which is green color. So it means that we look at one gene only. Can I see that? Okay, we look at one gene only. So in this case, we're going to involve the law, Mendelian law of segregation. Okay, this thing happens during the meiosis one, Mendelian's law of segregation. So a famous example in this cross, basically, if I have the heterozygous yellow seed cross with another heterozygous yellow seed, so I know that they're heterozygous, so it must big Y, small y for yellow, big Y, small y. And then, so this is the F1, okay, F1 phenotype. Then this is F1 genotype. So from F1 phenotype and F1 genotype, you're going to have the gamete genotype. So because of law of segregations, after the meiosis one, we're going to segregate the chromosome, the homologous chromosome, into two cells. So in this case, you're going to have half big Y, half small y. Same goes to here, half big Y, half small y. So when any fertilization takes place, in this case, you can draw the Punnett square or the genetic diagrams, then you're going to get F2 phenotypes and genotypes. So we're going to have here is big Y cross with big Y. We have big Y, big Y cross with small y, big Y cross with small y, and small y cross with small y. And each of them, you're going to have 1/4 chance. So you consider everything here, then the ratio for the phenotypes here. So this is yellow, because dominant allele must affect recessive allele, and this is green. So therefore, what you can see here is if I get the ratio of yellow to green, you will see that 3 times yellow appear and each time is 1/4. And for green to appear is 1 time only, red is 1/4. So the ratio is 3:1. Okay, so in terms of constructions of the genetic diagrams, we have few things that we need to know. For F1 phenotypes, and okay, we always can check the question. I will say that. On question, you'll be able to get what is the F1 phenotype. And then from the question also, you need to figure out the genotypes. Okay, whether they're heterozygous or pure breeding. Okay, then gamete genotypes, okay, what you need to know here is, first thing, you need to have the probability. Second, you need to have the circle for the gamete genotypes. Okay, so this you must know. And then when it comes to the genetic diagrams, you must have 3 things. First, probability of each genotype. Then you must have the genotype and the corresponding phenotype. Can you see that? So this is the things that you need to know, the 3 things that you need to know. And then in terms of the ratio, this is what you have to do, includes the probability for the ratio. Okay, it's a probability. Okay, you must include in terms of ratio to show that you get this probability or this ratio based on the probability. Okay, so 3:1 we call as a typical ratio or typical phenotypic ratio for heterozygous cross with heterozygous in monohybrid cross. Are you clear? So if you cross heterozygous with heterozygous in monohybrid cross, this is the what we call this the phenotypic ratio 3:1 is a typical ratio. This is Mendel ratio. Okay, so what we're going to do later, we're going to see what are the cases that they're going to modify this ratio. Are you clear? So always remember 3 for dominant trait, 1 for the recessive, always. Okay, dominant and recessive. Okay, so if you look at the dihybrid cross, what's the typical ratio? Dihybrid cross, you do the same thing, then you get 9, 3, 3, 1. So what's mean dihybrid cross? It's a typical ratio. Okay, sorry, for typical ratio. Okay, for heterozygous cross with heterozygous in dihybrid cross. Now, in dihybrid cross, then what will happen here is, now what's mean dihybrid cross? We're going to look at these 2 genes. Let's say, for example, code for the seed color and another gene code for the seed structure. 2 genes. Are you clear? So in terms of Mendel's, then they involve the law, okay, of independent assortment. Are you clear? Involve this law of independent assortment. So with the law of independent assortments, then we have one term and conditions here. What's the term and condition? These 2 genes must located on 2 different chromosomes. Or in another word, say that they are not linked by 1 chromosome. They are not linked. Are you clear? They must located on the 2 different chromosomes. For example, two chromosomes. So I have the, let's say, for example, allele big A, allele small a. Then allele big B, allele small b. So located on 2 different chromosomes, then you're going to fulfill the law of independent assortments. Then you will be able to get 9, 3, 3, 1. 9 for dominant trait for 2 dominant traits. Okay, 2 dominant trait. 3 and 3 actually for 1 dominant trait. Okay, 1 here is for 2 recessive trait. Okay, so you get the ratio of 9, 3, 3, 1. So with this, I have explained and summarized what's meant by the monohybrid cross and also dihybrid cross. So today we're going into the topics where we're going to see the modifications on the phenotypic ratio. We won't fulfill 3:1. We won't fulfill 9, 3, 3, 1 anymore. Due to what? Due to a certain factors that we need to learn today. Okay. So we're going to start it with the first one called codominance. Okay, we're going to start the first one called codominance. So in terms of codominance, basically means that you're going to give us a heterozygous, a special type of heterozygous, because of both allele. They cannot, totally dominant compared to another one. So therefore, the genes now, both allele of the gene are equally expressed. Means that, let's say for example, if you look at this allele, like we have learned, yellow allele, dominant. this green allele. So whenever big Y and small y allele together, big Y allele always show the effect, because the effect of dominant allele masks the effect of the recessive allele. So that's why green cannot be shown. But in codominant case, both of them equally expressed. So it means that you're going to have both characteristics in one cell. Okay, so one of the famous example that we always look at is, one of the famous example we always look at it. Let's say you have IA and IB together. They are codominants. A code for blood group A, B code for blood group B. So when they're together, blood group become AB. So this is termed as the codominance because both of them equally express. Okay, and if equally express, the heterozygous here, when equally expressed, because we can detect, right? For example, blood group AB, we can detect. As long as we can detect, it becomes a special, a distinct group. So these distinct phenotypes in the heterozygous, we name them, if the disease, we name them as a carrier. Are you clear? Okay, we name them as a carrier. Okay, they may not show a symptom, or they may not show a very severe symptom, but they do carry the allele of that particular disease. Okay, so look at this. One example, sickle cell anemia. Okay, sickle cell anemia. So this allele in the beta polypeptide genes, you can see that there is a base substitution. Okay, from normal T now becomes A. Thymines is substituted by adenine. So therefore, if they form a different kind of the beta polypeptide. Okay, so we have two different of them. So one is HBA allele, another HBS. HBA code for normal beta polypeptide. HBS code for the sickle cell beta polypeptide. Okay, so now we need to draw a genetic diagram to show that heterozygous male and female. So heterozygous, basically HBA, HBS, having a child in F1 with sickle cell anemia. So in this case, my parental generation is no longer, homozygous. I didn't say it's a pure breeding. So in this case, both of them, they are carrier. So what's mean carrier? Carrier basically means that heterozygous. So you can see that in this case, HBA, HBS, HBA, HBS. Okay, sorry for my mistake here. This is HBS, this is HBS. So half of the chance of the gametes will have HBA, half of the chance will have HBS. So when you cross both of them together, HBS, HBA, you get a HBA, 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 HBS as a carrier, HBA, HBS as a carrier, HBS, HBS as the one that having sickle cell. Are you clear? Or sickle cell anemia in this case. Okay, so probability, you just need to multiply 1/2, multiply 1/2, get 1/4. Now we look at the phenotype. So if you check normal, so means that all beta polypeptide will be normal, no defective. In this case, you can see that if you check the beta polypeptide, 50% will be normal, the beta polypeptide, and 50% will be the abnormal beta polypeptide. So you can see that there is a distinct group already different from this normal group. So this group, we name them as a carrier. So it means that carrier become a phenotype. Are you clear? Later, don't confuse, later we have another one. They are carrier, but they don't belong to a new phenotype. This one is a new phenotype, okay, called carrier. And the last one, you can see that all beta polypeptides abnormal. So therefore, we have three phenotypes. One is normal, another one is carrier, another is sickle cells. Therefore, what is the phenotypic ratio? Normal to carrier to sickle cell anemia. So you can see that the ratio is 1:2:1, no longer 3:1. This is considered modifications to the monohybrid because one gene. It's considered as a monohybrid cross, but it's not following. This one is not following the Mendel. Okay, they don't follow the Mendel's ratio anymore. In this case, I get 1:2:1. Are you clear so far? Okay, so modification, no longer 3 to 1 already, because this carrier become one of the phenotypes. You can check their blood and blood cells, you realize that they have 50% normal and 50% abnormal beta hemoglobins. Okay. So in codominance, neither of the allele are dominant in this case with respect to others. So means that no matter how they are equally expressed, so therefore in heterozygous individual will express both phenotypes. Can you see that? If you follow this one, they have the phenotype of normal, they have phenotype of sickle cell, but this carrier will have the mixture of both of them, normal and also sickle cell. Are you clear? Normal and also sickle cells or abnormal beta globins. So another example in the codominance, a parental red homozygous flower and the parental white homozygous camellia flower will produce F1 offspring with the flower have the red and white spots. Okay? Sorry, red and white spots. So against codominance, if you look at it, red to spotted to white is in the ratio 1 to 2 to 1. Are you clear? You get 1 to 2 to 1 rather than 3 to 1. So this again another example, okay, of what we call it as the codominance. Okay. Any question regarding to the codominance? If no,